Hi, my name's Heather Farrington. I'm the zoology curator at the Cincinnati Museum Center. Today I'd like to share with you one of the projects going on in our DNA lab at Union Terminal, and this project focuses on genetic sexing in birds. In many bird species, it's very easy to tell the males from the females. This is known as sexual dimorphism. So it's two physical forms, one for male, one for female, within a species. You're probably familiar with some of the examples we have pictured here, the northern cardinals that probably come to your backyard feeders, mallard ducks that you've probably seen on local waterways, and if you've been to the zoo, you've probably seen the peacocks running around the grounds, which is an extreme example of sexual dimorphism. So in these species, the males are typically very brightly colored and boldly patterned, whereas the females are pretty drab, usually browns and grays, and not nearly as flashy as the males. But not all bird species are dimorphic. Many are monomorphic. For example, raptors or birds of prey and lots of shorebirds display sexual monomorphism, meaning the males and females look identical. Another example when it's not so easy to tell males from females are when birds are very young. So when you have birds that are still in the nest or still have juvenile plumage, it's very difficult to tell the males and females apart. So why do we care? Well, it's important to know how many males and females we have in a population to look at population growth potential. So the females are the only ones that are going to be laying eggs, and therefore the potential growth in a population is really dependent on them. Another example would be in captive breeding. If you're trying to captively breed animals, you want to make sure that you have males and females paired together, otherwise you won't get successful reproduction. Also, some species of birds have differences in behavior that are based on whether they're male or female. So our current research project actually focuses on two local monomorphic species. The first is the blue jay, that's a year-round resident in the area. You've probably seen those in your backyard. And the other is the white-throated sparrow, which is a winter resident that heads north into Canada to breed in the summer. So the research question we're looking into is, are there morphological measurements we can take in the field on these birds to determine if they're male or female? So we're actually doing this project in conjunction with a professor at Miami University named Dave Russell. And he runs a banding station where he can actually catch these birds, put federally issued identification bands on their legs, and while he has them, he's able to take morphological measurements, things like beak size, skull size and shape, foot and leg measurements, wing measurements, tail measurements. What he wants to know is there a morphological measurement that he can take when he has the bird in hand that will tell him if it's a male or female for these monomorphic species? And in order to do that, we need to provide him with the information on who the males and females are that he's captured. So to give you a background on the genetic basis of sex, in mammals, you're probably familiar with females being XX and males being XY. But in birds, it's actually reversed. So the males are XX and the females are XY. So that's kind of an interesting difference. And because of this difference, instead of referring to bird sex chromosomes as X and Y, they're actually referred to as the Z and W chromosomes. So males in birds are ZZ and females are ZW. And whichever sex has the different sex chromosomes, so the XY or the ZW, are known as the heterogametic sex. So hetero being different, so those are the heterogametic sexes. It's males in mammals and females in birds. So the differences between the Z and W chromosome, the W chromosome is much smaller. And when you have genes that are found on both the Z and the W chromosome, the copy on the W chromosome is usually smaller. And that's because the introns, or the non-coding regions of the gene, have been removed. So what you typically get is a smaller version of the same gene on the W chromosome compared to the Z. So what we can do in these genetic sexing tests is we can actually copy or amplify that particular gene on both chromosomes and see if we get two different size products. And we can run those on a gel electrophoresis, which I'll talk about in just a minute. And what we'll see is two bands for females and a single band for male. And all that means is we've copied two different size versions of the same gene from the two different chromosomes. So how do we get the bird DNA? So when these birds are banded, we collect two tail feathers. And what we really want is the base of the feather where the skin cells are concentrated. So if you've ever seen a crime drama where they need a DNA sample from someone, they'll pull a hair because they want the root. They want those skin cells that are attached to the hair, or in this case, the feathers, where the DNA is going to be concentrated. So we actually cut the tip of the feather off and extract the DNA from that. So anytime you do genetic research, the very first step is always isolating and purifying the DNA from the sample. So we need to get the DNA out of the cells that contain it. So basically what we do is we take a tissue sample, in this case the tips of our feathers, and we soak that in a strong detergent that breaks down cell membranes. So cell membranes are mostly fat. So just like when you use detergent to do your dishes, it's going to break down that cell membrane and let out all of the stuff that's inside. And then we also add proteinase K, which is a protein that breaks down, or an enzyme that breaks down proteins in the cells. So then what we need to do is go through a process of purifying and isolating that DNA from the rest of the cell parts that we don't want. So isolating the DNA from all of those fats and proteins that we've broken down from the cell. And what we're left with is a purified DNA sample that's basically your double helix with your sequence of A's, G's, C's, and T's that make up our genetic code. So once we have our DNA sample isolated and purified, we use a process called polymerase chain reaction, or PCR, to copy those particular regions of the DNA that we're interested in. So basically, we take our DNA and put it in a reaction with primers, which are actually the small pieces of DNA that are going to help define what we're copying out of the genome. Your dNTPs, which are your A's, G's, C's, and T's. So anytime you want to make more copies of DNA, you need those building blocks. You need the A's, G's, C's, and T's to make more DNA strands. And we also have a polymerase enzyme, which actually does the copying for us. And what we do is we put that reaction into what's called a thermocycler, which raises and lowers the temperature of our reaction over and over and over again. And what happens in a PCR reaction, if we pretend these little blue equal sign shapes are double-stranded DNA. So if we pretend this is double-stranded DNA, we raise the temperature in our PCR, and DNA will actually melt at a high temperature. And melting of DNA means that the two strands of the double helix pull apart. And after we melt the DNA and pull those double strands into single strands, we reduce the temperature, and the enzyme will come along and copy the piece of DNA that the primers designate. And then we raise the temperature again, and those two double-stranded DNA fragments become 4 single-stranded DNA fragments. We lower the temperature again. The enzyme comes along and copies those single strands to double strands, and we repeat the process over and over and over again. And every time we raise and lower the temperature, and this reaction goes through a cycle, we get a doubling of the number of copies of our DNA. So we go from 1 copy to 2 to 4 to 8 to 16 to 32 and so on until we get lots and lots and lots of copies of this particular gene or region of DNA that we're interested in. So as I said, we do this for many, many cycles. So we typically run these reactions for 30 to 40 cycles. And you can see in this little image here, we have glass vials that have red beads in them. And we start with one bead in the tube on the left, and every tube has double the number of beads. And you can see it doesn't take very long once you get a few copies to really take off. And that exponential growth is what gives us so many copies at the end of a PCR reaction. So after a typical 30-cycle reaction that takes a few hours, we would have over 1 billion copies of DNA from a single starting strand. So after we have our PCR product, we need to see what we got. Did we get 2 products, one from the W and one from the Z chromosome, or did we just get one, meaning we had 2 Z chromosomes present? So to do a gel, we basically make a gel almost like Jell-O. We dissolve a powder in a liquid and let it solidify. And that gel also contains ethidium bromide, which I'll talk about in just a second. We load the samples into the gel, and we basically put it into a chamber that contains salt water, and we hook it up to a battery so that there's a positive side and a negative side, and an electrical charge runs through that saltwater solution that we have our gel submerged in. And as so DNA is negatively charged, so it will run toward the positive side of the gel. And as it moves through the gel, it picks up ethidium bromide, which is a molecule that will glow under UV light. So once the gel is run, we can put it on a UV light platform, and we can actually see the bands of DNA on our gel. So to give you a little animation of what that looks like, we have a gel with wells that we'll load our sample into. And then once all the wells are filled, we put it into our chamber and put the electric current across. So we have a negative end and a positive end. The DNA will move toward the positive charge. And what happens is smaller fragments of DNA will move through the gel more easily than larger ones. So the smaller pieces of DNA will move faster. So just like a small child weaving their way through a crowd much more easily than an adult. So the fragments will move. Once we're finished, we expose it to UV light. And the ethidium bromide will make our bands glow so we can see them. So here's your pop quiz for today's presentation. Who are the females on this gel? So this is actually a gel we ran in early February. What we were doing was looking for positive controls that we could use for our test. So anytime you run a genetic sexing test in the lab, you want to have a positive control so that you know what a male and female sample would look like at the end of your test. So we actually collected 11 Blue Jay tissue samples from the frozen tissue collection at the museum and ran the PCR for the CHD gene, which is a gene that's found on both the W and Z chromosomes. And we went through the process that I just described, and this was the result that we got. So over here on the right is actually what's called a size standard. So each of these bands is a DNA fragment of a known size that we can compare our bands to. These are our 11 samples, and this empty spot here is actually a negative control so that we can test and make sure that none of our reagents are contaminated with DNA. So if you look at this gel, remember we're looking for 2 bands for females and 1 band for males. So who on this picture represents our females? And if you said these 2 samples right here, you would be correct, A+ for the day. So these samples we're actually going to be using for our positive controls on this assay. So when we run the samples that were collected in the field by Dave, we'll be able to compare those results to known males and females from our tissue collection at the museum. So thank you so much for joining me today. I hope you got a lot out of this talk, and we look forward to seeing you at the DNA lab when Union Terminal reopens. Until then, please stay safe and healthy. And if you're willing to make a donation to the museum, either through renewing your membership or purchasing a membership for the first time, or donating to our COVID-19 fund, we would greatly appreciate it. So until next time, thanks for joining me, and we'll see you soon.